Yes, so dear participants of Euro-BioImaging, dear organizers, it's a great pleasure for me to present our scientific project and innovative technological solutions. So my name is Diana Mechtcheriakova. I'm docent at the Institute of Pathophysiology and Allergy Research of the Medical University of Vienna and the head of the research group Molecular Systems Biology and Pathophysiology. My presentation today is entitled Unlocking the Lymphoid Structures with Germinal Centers, Architectural Complexity, Functionality, and Clinical Relevance. So a few words about our group, Molecular Systems Biology and Pathophysiology. So our concept of the research is from systems biology to systems medicine, as part of the global concept of personalized medicine, and it's in line with the aphorism of philosopher Aristotle: the whole is greater than the sum of its parts. The approach is holistic for deciphering the complexity and follows the hierarchical flow from data to information to knowledge. The main research areas is systems biology approaches, which we developed for various research projects. And the main focus is given to B-cell biology, immuno-oncology, lymphoid structures, the EID upper-back-associated biological events, and cellular sphingolipid lysophosphatidylated system in immunity and cancer. So I'm also for quite a long time a head of tissue image cytometry unit at Austrian Bioimaging and the head of basic and translational research model at Austrian platform for personalized medicine. Today, the part of the scientific work which I'm going to show and discuss with you will focus on the biology of lymphoid structures. So these are highly organized structures with tight packing and interaction and unique functional cooperation of various immune cell types, including B-cell subsets, specialized follicular T helper cells, follicular dendritic cells. When fully developed, those structures contain germinal centers, and these are functional niche where B cells proliferate, differentiate, undergo affinity maturation, and switch the class of their antibodies. And activation-induced cytidine deaminase, EID, is a unique marker of functionally active germinal centers. So you see this staining here on the tonsil tissue. The outcome of GC reaction is production of plasma and memory B cells. So those lymphoid structures are located in secondary lymphoid organs, but additionally and importantly that they can be assembled at ectopic sites within chronically inflamed tissues, including the tumor site. And in this case, they are known as ectopic lymphoid structures, ELS, or tertiary lymphoid structures, TLS. So specifically to highlight is the clinical relevance of B cells and ELS in cancer. And it was demonstrated, and this I believe is among the breakthrough discoveries of last years, that the local presence of B cells and ectopic lymphoid structures associated with favorable patient prognosis in more than 10 cancer types. And to exemplify this is non-small cell lung cancer, breast cancer, and colorectal cancer with metastasis in the liver. So in respect of colorectal cancer with metastasis in the liver, we were pioneers to show the accumulation of B cells assembled in ectopic lymphoid structures with active germinal centers in the liver, and those ELS formed a ring around the metastasis. So we calculated the number of B cells and ELS precisely at the tumor liver border with the 0.5 millimeter areas directed in both directions to the tumor and to the liver. And this was done using quantitative tissue image cytometry. So this was our scientific intuition multiplied by the power of this technology. And we used the tissue fax-based computerized microscopy. So what we found that the higher magnitude of B cells and ectopic lymphoid structures associated with better clinical outcome in terms of survival. And what was amazing that this prognostic effect was superior to clinical pathological parameters. So we next hypothesized that the anti-tumor immune response at the metastatic site in the liver where actually ELS are biological, physiological foreigners are pre-instructed at the primary CRC site and or even at the non-tumorous colon mucosa with the natural isolated lymphoid structures known as ILS. So the study addressed the research question whether specific characteristic of lymphoid structures established at these three different tissue entities have an impact on the pathobiology of metastatic CRC. So to assess that, we established and developed a multimodular DICO strategy abbreviated from digital immune imaging to clinical outcome. So DICO model 1 starts from the immunostaining with the set of predefined markers and the marker combinations. And in this study, we use CD20, the general B-cell marker, AID for functionally active germinal centers, Ki67 for proliferating cells, CD27 for memory B cells and plasma cells, as well as T cell subsets, CD138 for plasma cells, and CD3 as general T cell marker. And those markers and combinations were applied for colon cancer specimens and tonsil tissues to address immune cell composition and activity of lymphoid structures. So the second DICO model is actually assessment of immunological imprint by quantitative tissue image cytometry. The stained specimens were acquired using automatic macroscopy-based tissue fax platform to get digital images. Then quantitative analysis of these digital images were done using HistoQuest and TissueQuest software packages. The lymphoid structures were defined as the objects, and this analysis is done on the single cell recognition, and the marker positive cell detection is going by a marker specific profile. So I'm very happy to say that we have this powerful technology represented by the TissueFax platform in our institute since 2008. So we have long-term collaboration with excellent tissue agnostic team. And in 2022, we became a center of excellence for quantitative digital microscopy. So using this powerful technology, just in this particular project, more than 700 tissue slides were analyzed from the colon cancer specimens and more than 120 tonsils. The outcome of such analysis is a transformation of tissue encrypted information into numerical values as variables. Anatomy and staining-based variables exported for each tissue and each patient, and variables are then used for alignment with clinical pathological parameters. So this is the full list of the 24 variables attributed to lymphoid structure characteristics, and this includes such parameters as number of lymphoid structures, size of lymphoid structures, cellular density, and the percentage of positive cells for each marker. So important question which we addressed is the clinical relevance of variables characterizing the immune phenotype of lymphoid structures. And intriguingly, what we found for NT tissue, strong prognostic power for the variables attributed to percentage of CD20 positive cells, Ki67 positive cells, as well as for their combination when calculated within the lymphoid structures. And this you can see from statistically significant stratification of patients into the lower and high-risk groups using the Kaplan-Meier estimates. So major discovery to highlight here that B-cell enriched and highly proliferative ILS located within non-tumorous colonic mucosa showed a strong prognostic effect in respect of survival of CRC-LM patients. So this in turn highlight that immunological imprint of lymphoid structures within normal colon encrypts the information about clinical outcome for patients with metastatic colorectal cancer. So what is now, what is next? We integrated to our systems biology-based multimodular analysis strategy. In addition to quantitative tissue image cytometry represented by TissueFax technology, and compendium-wide analysis of transcriptomic dataset represented by Gene Investigator tool, 2 additional modules, and this is spatial transcriptomics by 10x genomics using the Visium spatial gene expression, where the mRNA-seq is done directly in FFPE tissue sections, and IE in biomedical research as part of our new ongoing project, lymphoid structure miner, which is done in collaboration with IE specialists from Danube Private University in Austria. So we will utilize the power and synergy of 4 analytical models for unlocking further the germinal center biology, and we hope this will lead to nomination of novel biomarkers and targeting strategies. So with that, I would like to acknowledge all members of my MSPP group, especially for these results I have shown today. These are Anastasia Mechtcheriakova, Felicitas Munginast, and Anastasia Marchuk, as well as our great collaborative team, a multidisciplinary team for multidisciplinary studies and the funding agencies. Felicitas and Anastasia are now part of tissue agnostic team, and Anastasia will continue with her part of presentation for innovative technological solutions. So thank you for your attention.